So now we're going to discuss organelle DNA in Chapter 11. We'll first discuss the endosymbiote theory, then chloroplast DNA, cpDNA, and then mitochondrial DNA or mtDNA. So the endosymbiote theory states that these organelles, mitochondria and chloroplasts, evolved from free-living bacterial cells engulfed by eukaryotic cells. And this happened between 1 to 5 billion years ago. It's thought that mitochondria evolved first. Some evidence for the theory is that the present-day protists have endosymbiotic eubacteria, and the confirming evidence really is the DNA sequences of the organelle genomes indicate that they evolved from free-living bacteria. So, looks something like this: an anaerobic eukaryotic cell via endocytosis engulfs a bacterium, and instead of digesting it, the bacteria takes up forms a symbiotic relationship with the cell, and then evolves into the present-day mitochondria. And later on, a cell with mitochondria engulfs a cyanobacterium, a photosynthetic bacterium, and over time that evolves into modern-day chloroplasts. Here's those two slides, start to finish. So both these organelles exhibit maternal inheritance. They're always inherited from the maternal parent because they are in the egg cytoplasm. Both organelles are involved in the basic metabolic processes. As seen here, mitochondria are involved in aerobic metabolism, and of course chloroplasts are involved in photosynthesis. So chloroplast DNA tends to be a large circular molecule with and we know it evolved from photosynthetic bacteria called cyanobacteria based on the DNA sequence. Mitochondria tend to be a smaller circular molecule. For instance, in us humans, it's got 13 structural genes and 22 transfer RNA genes, and it evolved from a group of bacteria called purple bacteria. So here is a comparison of the two organelles. Chloroplasts tend to be a bit larger than mitochondria, although mitochondria can fuse to make long worm-like structures or small round vesicle-like structures as well. So most cells and organisms exhibit what's called homoplasmy for organelle genomes. That means all the mitochondrial DNA molecules inside your body are the same in every cell. That makes sense, right? Since we start off life as a zygote, a single cell, with all your mitochondria coming from your mother. So therefore, your mitochondrial genome should be the same as well. So here's a plant cell showing you plants have both mitochondria and chloroplasts. We'll discuss chloroplasts first. Chloroplast DNA, the DNA is supercoiled in the stroma, or the liquid filling of the chloroplast, as we can see there. Here's a map of the rice genome, Oryza sativa. So it's large relative to mitochondrial genomes, 134,000 base pairs. Blue regions are protein coding genes. The different color green regions are either transfer or ribosomal RNA genes, and the yellow are ORFs, or open reading frames. These are DNA sequences that appear to be like genes, but we haven't confirmed that they really are genes or not. We don't know of any product that they make, but it seems like they have start and stop codons and promoter regions and all that kind of stuff. This shows you the sizes of the chloroplast genomes in various organisms. So as small as 119,000 in the black pine tree, and as large as 191,000 in a species of red algae. Moving on into the mitochondrial genome, of course, here's an animal cell showing you the mitochondria in the cytoplasm. And we see that the mitochondria live inside the matrix, which is the inner compartment inside the inner membrane there. The little black dots are ribosomes. Mitochondria reproduce by binary fission like bacteria do. This shows the process in liver cells. Turning from left to right. Here is a euglena, a protist that is has both mitochondria and chloroplasts. The big orangey thing there is the nucleus of the cell, and the mitochondria here have been stained green, and within them the mitochondrial DNA are the little yellow dots. You can see there's a lot of them in there. in that one cell. Those little yellow dots, each one is a mitochondrial DNA molecule. Here's a map of the human mitochondrial genome. Just under 17,000 base pairs, 13 structural genes and 22 transfer RNA genes. So those things that appear to be amino acid name abbreviations, like at the top, phenylalanine, valine, proline, etc. Those are the transfer RNA genes. This shows at the bottom here the human mitochondrial DNA genome or molecule, all negatively supercoiled, like it resides in the matrix of the mitochondrion. But not all organisms have small mitochondrial genomes. Here's a map from yeast. It's got 78,000 base pairs in its mitochondrial genome and a lot of non-coding regions. See these gaps between the genes there seem to be non-coding DNA. Again, protein genes are in blue, and ribosomal RNA and transfer RNA genes are in various shades of green. This shows you mitochondrial genome sizes of various organisms. And in most vertebrates, it's somewhere between 16 and 20,000 base pairs, as we can see here. But in some plants, it's really, really huge. Dr. Rentz tells me that people who study plants use chloroplast DNA to study plant evolution and not mitochondrial DNA, because it's pretty messy. This shows that some of the codons in the mitochondrial genome are different than the universal genetic code. For instance, the first one, UGA, of course, is a stop codon, but it codes for tryptophan in mitochondrial genomes. AUA is isoleucine in the universal code, but methionine in the mitochondrial genomes. And AGA is arginine in the universal code, but it's a stop codon in vertebrates, and a serine codon in Drosophila, but it's arginine in yeast. So, now, mitochondrial DNA has been studied a lot in animals, particularly for evolutionary studies, and it's got several reasons that it's quite convenient to study. One is its maternal inheritance, so we always know where it comes from. It's haploid. Only one copy in the organism does not undergo recombination, so it's inherited clonally. So everybody, our mitochondrial genomes are identical to those in our mother, unless there's been a mutation. It's a circular molecule, which makes genetic mapping convenient, and it's got a pretty high evolutionary rate, and that makes it good for studying population genetics and evolutionary relationships of closely related species, like things in the same genus or family or whatever. Like has been done here to determine that the domestic donkey descended from African wild asses, which are these creatures. Equus africanus is the African wild ass. And then this shows a phylogeny of that with the donkeys and the African wild asses in that top group, Asiatic half asses in the middle group, and then domestic horses down there at the bottom there, Equus caballos. So these phylogenies are hypotheses of the evolutionary relationships of these organisms, and this is based on the sequences of their mitochondrial genomes. So the reason that the people that study plants don't use mitochondrial DNA is in plants it can undergo recombination, and it's pretty difficult and messy to study. But the mitochondrial genome can undergo various mutations, and they can have deleterious phenotypic effects. So a deletion in the mitochondrial DNA molecule can cause what's called Kearns-Sayre syndrome. And symptoms of this include hearing and vision loss, heart defects. However, children don't show the symptoms. They appear as the person ages, all because you're missing a little part of your mitochondrial DNA molecule. Another one is a mutation that interferes with translation in one transfer RNA gene, only one of them, and it causes myoclonic epilepsy and ragged red fiber disease, which is called MRF. Symptoms of this include mitochondria lacking Christie, those inner folds of the mitochondrion, but people with this have abnormal muscle fibers and deafness, dementia, and seizures. So it's very important that your mitochondrial DNA be complete and normal for you to have a normal phenotype. And then there are mutations in yeast as well. one called a petite mutation is a deletion that makes the cells unable to carry out aerobic cellular respiration. So the colonies of these yeast are really tiny, as we can see here in this bottom panel on the right, where the normal colonies are rather large on the Petri dish. And that is all I have for organelle genes or genomes.